Assalamu alaikum wa rahmatullahi wa barakatuh, brothers and sisters. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. Inshallah ta'ala, students will be coming in shortly for BIOC17 today, inshallah ta'ala, going into week 7. So bear with me, inshallah ta'ala, as we move further into today's practical and scientific students, and Allah subhanahu wa ta'ala make this the best of all tests. Ameen ya Rabb.
Sorry. Always. How's it going? Yeah, we have a class. We just have to do it. You know, I remember. Yeah. It's like a lot of my friends and I think they will get a great for us. So they're doing it. Yeah. And like, it's just like the other. And she's it. And she's yes. Yeah. Like the family really even me one other girl the other two haven't been helped. One thing left? No, one from like the class. The discussion. Oh yeah. I know. Oh. Yeah, she's like, I don't know how long it's supposed to like. For yeah. Oh yeah. So that the UV rays. We have a no, there's just having that. Oh, it's not in the left and I thought it was more left. I hope not, man. I don't know what to. Wait, you're gonna like once you oh I want to take it. Oh. And I think we do the and then we can start. These are the first experiment. You're another shot as well. No, but yeah, but oh, doors. There's that.
No, those are not our numbers. Yeah. Give me one minute. Okay, actually I could go. This one. Right up today. This is the- Um, continue. Proceed. You'll see. Can you send me some more? One of these. But, okay.
So I could totally get the
I actually posted it on the map, so if you see your inbox, you should be able to notice the results interpreting other people. This is this the top. Yeah, it's like the exercise. So if you can try to visualize it for upcoming popular economy. So just a quick question. Is Amisha here? No? Okay. Okay. Is she planning to come if you know? Okay. So I'm just making see if you guys can access. Okay, everyone. I don't know if you guys can see it. Um, This is good news. Both screens are working. So, congrats everyone. You've made it not only 60% into this class, into our practicals together, but you're in the last 4 weeks, so in the final stretch, really, of BIOC17. So, congrats, folks. I know many of you are also wondering about the plaque assay. FLR, it takes a bit of time in terms of marking, but I do hope, inshallah, with the results and discussion comments that I've provided before and in your reports, that hopefully you will implement them for your second lab report, which will be on the catalase test and your results from this week. So we'll talk about a few things. Also, for those that don't know, actually, maybe I'll talk about it afterwards, but we're doing some analysis today for the chemical germicide. So if you haven't looked on Quirkus, have a look at those results, but you'll be presenting them today in about 10-ish for 10 minutes per group to discuss your results. And I'll talk about that in due time. But what I do want to focus on is a bit more so on your your first two exercises for today, and that is the lethal effect of UV and UV radiation, respectively. Now, if you have a look, and I think I do want to see for timing. I think I'll go through briefly today's exercises and what you have at your disposal. For exercise 2, because it's a bit straightforward with the result that you have, there are so that being said, groups of for context, this is where exercise 2, if they say time here. The plates that we have MH plates like the ones that we used last week, you'll be using them to follow up with the antimicrobial susceptibility test. We'll talk about that in due time. But for exercise 1, you have the plates already on your bench. So just bear that in mind as groups of 3 to 4. In case there are a group of 2, that also suffices. But what I will say, actually, you're a group of 3, right? Perfect. Okay. There's a group of 2. Let me know. Vishwa, you're in this group, right? Just a group for perfect. Okay. So let's go straight ahead. Okay. So really, once again, if we have a look as to what we're doing for today, a lot of it deals with UV. If I just scroll shortly onto the lab manual, you'll see further details, but we're dealing with E. coli and Staphylococcus epidermidis. Can I get a recap as to what makes them different from each other, from you? Okay. Okay. And so which one would have a thicker peptidoglycan layer? That's correct. Good. Which one would have a thicker peptidoglycan layer? Mhm. Sorry? This was the very last part. The retention? That's correct. So, well, something I want to point out, and then maybe someone else can answer this, is what can you hypothesize or predict then would be the effect of UV on either, or which one would have perhaps a more pronounced effect following UV? Yes. Why? Think about it. Think about it. Yes. Think about it.
And it doesn't have the 2 numbers.
And that's pretty much what I hope many of us will predict today. But we won't really see much of the results because the results take time. But hopefully next week you guys will be able to appreciate this and hopefully derive them for your post lab. B subtilis now, maybe with a show of other hands. What makes it different, or what's the difference between them at 24 hours and 7 days? Different set of hands. I see your hand. Any other hands? Actually, I'll go. There's actually a difference in this regard because technically 24 hours is like from the past we did 24 hours versus 48 hours, right? There was a particular reason for endospore staining. In this case, though, technically after 7 days, the endospores would have been released or essentially outside of the bacterium. So that would essentially mean then that the endospores themselves would not have the same function within the bacteria or essentially be subtilis. And then technically the UV's effects would probably be more pronounced at 7 days. 24 hours is a good point, vegetative as well for the bacterium themselves, but essentially the endospores would be likely of the ones of essentially produced still intact within the bacterium and thus likely would be more resistant to UV light. And here, for example, shown with UVC light. I just want you guys to also bear these questions in mind, not just for your lab exam, but for extensions of it in your lab exam. For example, repair mechanisms, which are often talked or thought about. So skip that final question. Actually, let's go straight. I'll go straight to the next slide. So exercise 2 for today, we have 7 different conditions. Group 1 will be tasked with, and let me just confirm. So group 1. Is it okay to see that if you can join their group? Okay. Reason being is because group 1 will have to do 2 conditions, and that's conditions 1 to 2. And then every other group will do the rest. So that would be group 2 doing condition 3, group 3 doing condition 4, group 4 doing condition 5, and then 6, and then 7. Does that make sense, folks, by the way? What you need to do is just like what we've done before, label your plates. Bear in mind with the timings for exposure of UV that you not only, of course, mask appropriately your different plates, but importantly as well, you are bearing in mind that you've essentially labeled and timed appropriately the plates that you are using for your group and based on the specific condition that I've just mentioned. The mutagenic effects, briefly, this isn't something that we may see today, but down the road, you may be able to see different colors of colonies. One, if it's, let's say, obscurely different, like if you have, I'm just going to draw it on PowerPoint. If you have, let's say, I don't think I can actually draw, but the idea is that if you, let's say from the plate that you just saw there, let's say this is your plate, and you have some that are red, which is expected, right? And then you have some that are pale, right? Which are actually the mutagenic ones, following UV light. But if you have ones that are more, like, abnormally different, right? They're likely is the case that you have cross-contamination. So just bear that in mind, folks, because especially, I mean, yes, we're dealing with UV. You'll see the different light canisters on either side. But you just want to bear in mind that you obviously maintain aseptic technique prior to this and even following these steps as well. Any questions for exercise 1 and 2? No. Well, sorry, actually, let me just go back. Yeah, there was another. You have each group should do all 7. Wait a sec. Why is it? Yeah. Yes. So this is what I actually wanted to go briefly into because the slides didn't show it. In case those that weren't aware, and great question, Rahma. Because yes, sorry, group 1 will essentially be focusing on both conditions. And actually, let me just cancel it. Let me show. It didn't disconnect. Okay, I'll tell you guys then. For exercise 1, where you have the plates on your benches, the plates, the 4 sets of plates per time point there. I want you to bear in mind, group 1, okay, maybe to make it easier, group 1, all groups will be using instead of 4 plates. You label them accordingly, right? For the different states that you have, for the conditions that I mentioned. But you essentially use the correct one, label them accordingly. The idea is that we go back, we will give you more contextual thoughts or ideas. Each of your groups will be doing 1 or 2 different exposure times. So bear that in mind. Someone has to have a timer on. But essentially, group 1 will be doing it without UV as a control, negative control, and at 5 minutes. And pretty much, once again, the lights for UV are down there on either side. I'll start on the UV soon, but I just want you to bear in mind that you guys, you can mask in advance for exercise 2 when it's time. But essentially, just bear in mind that you don't, regularly keep your hands in place with the UV. So that way you don't, actually, I don't think you guys have masks, right? For your hands like gloves. It would be nice to do that. But as an idea, so these are the masks. We'll focus on that. And this year, and then the upcoming 20 and 25 and 30 all the way at the end. Just bear in mind, not only the content, but the different cultures that each group has 5 different groups, if I'm not mistaken, with the cultures, Staphylococcus, epidermidis, and coli. Each group will just take 3 vials, and what that means is that essentially, when you're doing like what we've done before, like last week, you only need to take a finite amount and half of this to each 2, and that way you should have 2.5 essentially aliquots of or portions of saline into each of those tubes to readily gain a concentrated mixture of bacteria. But usually, once again, just to go over this, each group, based on the time points I mentioned earlier, will be essentially taking those 4 quadruplet plates and essentially labeling them with the age and name of the bacterium. You can split it in half to have A and B, as we've seen from your flow charts. And I didn't mention this, but results, by the way, for post labs, please make sure that they're descriptive with your figure captions, as I want to make sure that you guys are being as detailed as possible for, inshallah, future weeks. Please bear that in mind, folks. But briefly, inoculating, once again, your plates with each bacterial culture, and then essentially you'll have half of your plate, either A or B, essentially used as your as part of your mask. I don't know if it's provided further details. Yeah, so that's expected. I'm trying to see if there's anything else. Yes, so bear in mind that essentially you have the specific time points that should be labeled on those tubes. If you don't, let me know. But hopefully you have them for Staphylococcus epidermidis, B. subtilis, and E. coli. And so just bear in mind the different time points that are already presented for you. But you will use them and essentially follow spread plating as we've done before. And then thereafter, essentially make turns, 1/3 turns each time. And then hopefully you can repeat these steps and then place a paper towel on the bench next to the UV lamp to soak it up with 70% ethanol. You want to do that as it helps sterilize the bench top as well? Yes. Not, you don't necessarily. Yeah, it doesn't say that you have to. No, I wouldn't technically. Reason being is because you're gonna have to do aseptic technique, which means you'd flame your swab accordingly. So I wouldn't redo it. Follow pretty much what we did last week. Pretty much, actually, it's still here. I'll lift this up. But it's very similar to what we did with the Antimicrobial Susceptibility Test, which tells you what to see today. But the idea is that you just make 1/3 turns and swab accordingly with your bacterium. That's why it's very important, though, just to bear in mind that one, you don't break the agar, but two, for the tubes that you have, the 4 tubes that you'll be using today, make sure that they're concentrated, not overly diluted. Only half of the saline or like you don't have to overdo it essentially. And then let's see, at the end of exposure, let's see, range here. At the end of exposure time, extreme UV, replace lids immediately. Yes. So this is another thing. I realize it's not in your slides, but let me just show you guys. You guys can see it. It's I think I'll okay. Okay, it's working. So okay, briefly though, just wanted to highlight this. You're gonna be taking out, so follow the steps that you see from your manual and your flow chart, but importantly, when you're at the stage of adding, like when you're incubating your plates under UV, let me know accordingly, but we can begin now. Before I answer, wait, it's not showing. Okay. That's okay. If it doesn't show, I'll do things orally. I don't mind. If you guys also need help, let me know. One last time though. Now, in case folks don't understand this, when you're actually adding the UV, when you're placing your plates underneath, bear in mind that you not only have essentially done all steps for each of your 4 plates, having them arranged accordingly because there's only a finite amount of space on the lamp. The other thing that you want to bear in mind is that the paper towel is essentially placed soaked with 70% ethanol, and you guys should have eyewear. I want to know if we can get things for you guys at the end of exposure time. Read your Move the lids and place them open side down. Yes, so that's what I wanted to say. Pretty much, once again, group 1, 0, 5 minutes. Group 2, 10 minutes. Group 3, 15 minutes. Group 4, 20 minutes. 5, 25. And then 6, 30 minutes. Is that clear? Any questions? No questions? Okay, you guys can get straight ahead. I'm gonna also turn on the gas, so you just have to make sure 70% ethanol and this ball is and for the next half hour, for 40 minutes. So I see there are other things here. If your group hasn't taken them, please take the place. And so that way you can continue to do things as efficiently as possible. Work as pairs, work as trios, but just try to make it as coordinated as possible. Um,
Oh. Yes, sir. Wait, sorry, what?
yes. Yes, let's see. Sorry?
Yeah, yeah. And then you like
So I'm trying to split up these two four places. Try to do them in the next 10-ish minutes. I'll try to see if I can get you guys a timer as well. But it's 9:40, so if anyone has a clock with them, a timer, they could accordingly try to manage to complete the next set of plates in the next 10 minutes. Try to do them as soon as you can. You also have three sets of saline water. So include them essentially when you're diluting. Remember, only a finite amount is needed for each tube. So for each of the tube folks, please bear this in mind. For your agar plates, agar tubes, your solid agar block, please make sure that the amount of saline that you're adding in is only half of the tube. So go slow if you need to, but it's important that you don't overdo it. Otherwise, it's going to be overly diluted, and then the results may be poor. So bear that in mind. Also, bear in mind, aseptic technique, Bunsen burners should be on. Flame your lids accordingly. Make sure your plates are also labeled, as we talked about earlier. Practical exercise, the E. coli or B. subtilis or Staphylococcus epidermidis with the time point as well. So just bear those details in mind, including the dates. Bunsen burners should be on for all groups that have already started to use their bacterium. Please bear that in mind. Also, make sure, please, that your Bunsen burners are not too high up. If there's a smell of smoke, definitely it likely means that there are too many that are too high, or at least one that is. Just bear that in mind. You guys have a clean one, right? This one? I can. Oh, I read this. So, like, we're at the point where you stop, and you'll see the either halfway into your liquid, liquid or in the nuclear drug scientists of the product. So either way, just make sure that it's concentrated. Try to concentrate as much as you can without putting people.
Yes, there's already three paper towels.
No, I think we have
to do it ourselves. And we do it on the test, right?
Yeah, that's okay.
We have one. Oh, that was not quite. Is this like? Oh, okay, okay. Oh, I think this is what? Yeah. Yeah, you just have to. Yeah, yeah.
That's honestly a good setup for making the paper towels and essentially erasing it out of existence. Have a look at that slide. These are soap, they're not literally soap, but if you open the lid, it's dry. Very nice for these opening of the lid. Just bear in mind that especially when you have the paper towels that you definitely but when you that is simply
It's here. I think let me know when you guys are. And it was like all the and it was just one of us. I think that
I think we can here if you have that are on the longer time points, maybe should I ask how many of you have completed your plates or so this kind of research. All groups have done it. This is good for every group. Not at all are reading. The issue is that only really two groups really go at a time. So I would say, based on the groups that have already placed their sheets forward with new. That's our two things, folks. Bear in mind that you're wearing the goggles when you're actually using the UV. Another pointer is that someone needs to have a timer. Very important because that way we can coordinate who's doing that and hopefully complete the rest of today's task completely. Bear that in mind. I just want to make sure someone has a timer so that we don't lose time accordingly. Yes.
Take the lids off and put them face down. That's talking about a plate. You're taking a plate, the lid off and the subject.
If your plates are not actually at the edges exposed to UV, you need to shut it off and move it closer. Because otherwise, I noticed that you may actually not get strong results if you have the far from the edges. To the edges. So, well, I just want to clarify something for exercise 2. You all have 7 plates. In the meantime, for those that aren't aware, those 7 plates you'll be doing as a group. So to recall, exercise 1, you're all doing a particular condition, essentially, at a specific time point, right? Now, for exercise 2, you have 7 different plates. All of you will, each group will be doing all 7. So bear that in mind. You should have them all on your bench tops, and you can just label them accordingly. Because we have finite space. By the way, does that answer your question? Yeah. Any questions regarding exercise 2?
And so that actually is going to answer the upcoming question from Eunice as well for next steps. Because obviously, as we wait for exercise 2 or exercise 1 to be done, there's things that we could do in the meantime. Briefly, let's see if I can get this back. Sorry, you have a question. Are you sure? Okay. So, no.
So for essentially last week's practical, where we dealt with the antimicrobial susceptibility test, we're going to be talking about that in due time. The issue I'm having is that it's not showing on screen. So I'll talk about them and hopefully not answer your question. Pretty much, you recall your results. We had, or at least recall the methods. You had two plates with 5 different sections for the different types of antibiotics we use. And so the plan is that you're going to be now taking from those that have zones of inhibition, i.e. circles. Let's say, for example, this is your plate, which actually connects us back to previous practicals because essentially it comes down to subculturing, which is the techniques that you do in advanced courses, but also in grad school. So that's just a separate point. Now, recall it had 5 different antibiotics: ampicillin, penicillin, streptomycin, tetracycline, and chloramphenicol. Now, bear in mind, now you're going to see for the different plates, the different bacterium that you have, E. coli and Staphylococcus epidermidis. I'm just going to put. Bear in mind that you'll see results, right? These are your discs. But you may see results, some of which have a zone of inhibition, meaning that they appear to have like a big halo or circle surrounding that disc. Some of them may have it, some of them may not. Some of them may also have within that halo some daggered signs indicating a level of resistance. So briefly, what I will say is that for those that you see that show zones of inhibition, have colonies provided, you're going to actually subculture them into these other plates. You have 2 other MH plates, and essentially, if you want, you can make the same 5 sections and then essentially do what we've done before in terms of spread plating. You can use your inoculating loop and then essentially add them into, and actually there is specific details. We just don't have the projector on. So let's, let me just give you, let's see if it's going to show up. Okay, I don't think it's going to show up. So I'll just scroll down and just provide you the details accordingly. If you don't have it in your notes, then it's a good time to just take this into account. Briefly. So we're going back to week 6. If you need support on this, there's also a manual here. If you scroll, go back into lab 2 for its procedure, either in your manual or in your notebook. You want to just bear in mind that we're going to be subculturing the plates. And so what you want to bear in mind is that you start with a positive control. Remember aseptic technique, folks, very important, especially now because you're subculturing, meaning that essentially you are aiming to get a pure culture of your colonies from specific zones of inhibition, or at least from the specific sections from your previous plates. So bear that in mind. Bunsen burner flames should be on, and then you could follow aseptic technique accordingly. You can use a sterile cotton swab, and this is interesting because I don't know if you guys have extra saline. Yes. So essentially in that last, okay, I'll just give you guys what you need for now, for those that are waiting. Those groups that are done, let others know from your bench spots so that way they can accordingly move forward into the time for today.
Yeah.
And then when you're done, basically label accordingly. And then they will be able to essentially have. So Daniel, can you do that, or?
Daniel just calls IT if it's not coming on. The computer, not the projector. Okay. Mohammed, it's gonna show up from the desktop. It's going to restart. It's on. Start.
Okay,
Okay.
perfect.
So you just have to put the PowerPoint on the desktop.
Okay.
Because you haven't hooked up your laptop. Have you?
But in the past, it works out.
Okay, well, just let me. Is it a VGA or a HDMI?
So what you're gonna do now, on you get up the digital phone, make sure that you label for the evidence base the different sections. Make sure as well, based on the one that you have in specific place. Make sure as well that you use the sterile tube that you have. You can use the funnel swab, plug it in, and then take out essentially the given column from where the zones of inhibition are outside of them. Meaning, like, if you see a section that has zone of inhibition,
You'll have to be off the podium because your laptop.
okay. Thank you so much.
Now it'll take a while to reset. It's not recognizing your connection to your HDMI. Okay. So.
I appreciate it. Okay. Yeah. No. Okay. So briefly, folks, I'm just going to share what you need to do. So for those that are waiting, once again, I see that there is space. So perfect. Great job. Group 5 going in. Group that are here. Are you guys almost done for time? And what? Okay. When your group is done, make sure you switch off the UV. Very important. I don't want you guys to get your hands exposed, but then turn it back on thereafter. Now. So let me just briefly go through your next steps. For the antimicrobial susceptibility test results, you have your zones of inhibition for specific antibiotic. And so your job is the following. Let's see if I can get this back on.
Mm. No. Can I just get everyone's attention briefly for now actually, if your group is done and you have the opportunity to essentially move to exercise 2, I would recommend doing so because essentially, yes, there's a specific number of conditions that if you have a look at slide 3, even from your manual, or sorry, from the PowerPoint slides, the idea is that you're doing all 7 and really that they're in a range of 0 to 3 minutes. So it's very limited in timing. But in the meantime, you could start getting yourself ready for the controls that you'll be using. And so just bear that in mind. The second portion, or actually, for exercise 2, who can get their plates ready in the meantime? Because there are plates that you can do. And let me give you context. Plates 6 and 7 don't require UV. And essentially, when it says having them in dark, you're just either placing a paper towel or something solid on top of that plate to essentially support its incubation. And that'll be temporary, but the point is that I just want you to be able to make use of all time that you have today before we move further on. And then as I move further now, with the timing that we have, let me just say for exercise 2, once again, let's say this is your plate, right? The one, either of them, for E. coli, Staphylococcus epidermidis. Make sure that you label the other 2 plates that are shown with MH, like the Muller-Hinton agar plates, solid agar plates. Those same plates you're going to be using, but now you're going to be self-culturing sections that have a zone of inhibition using sterile cotton swab dipped into the saline water or saline solution. And then you're going to essentially take colonies from that section that are outside of the zone of inhibition. And then within that other plate that you've labeled accordingly, you're going to essentially dip or essentially take up that aliquot and then spread it across that section. The idea is that you want to be able to see if you can grow off colonies from there and hopefully see the results that they're in of hopefully single colonies, if not like a smear. But you definitely want to make sure you're following aseptic technique as you open up this plate, right? Because you want to make sure that as it's close by or adjacent to your Bunsen burner, that you don't run the risk of other microbes, let's say for example like this, that plop into your section or grow post incubation. So just bear that in mind. Once again, essentially you have both plates and then essentially follow from what I've shown here to answer any upcoming questions. So that way we can make full use of our time for the lab. Laiba, was there another question that you had, or was that the one? Okay, any questions regarding exercise 2? Or sorry, post-lab or last week's exercise 2? Yeah. Yes.
This with forceps and then swab in the cell.
So I saw as well, that is actually what I said initially, but the issue is, if I'm not mistaken, saying you do it outside. Yeah, it's saying outside the zone of inhibition. For the positive control, yes. Let me see if it shows. Okay, perfect. So I was right before. You're also correct. So we're all correct. What I mean is to clarify the following. when you're doing essentially your inoculations, bear in mind that rather than, and for context, you want to actually get the resistant colonies, right? So that way you'll be hopefully able to derive essentially the colonies that are resistant to the specific antibiotics of interest. So it's very simple. You're just repeating the steps. The one that you're taking from outside is pretty much your positive control, which isn't too much of an issue because you already have the bacterium available, pretty much from your place to start off. But it's important to point out the following, that you're doing this from the sections, as we can see here, there's 3 sections of zones of inhibition. So for context, for those that are treated with those antibiotics, take it from inside your zone of inhibition, and then essentially smear across accordingly. Follow aseptic technique. For those that are doing it from the positive control, do it from outside the zone of inhibition. You repeat the same process. You have a sterile swab, plunge it into a saline tube, and then accordingly take out, essentially smear from your colonies that are already present. Here, for example, from a section, to essentially blot it in or take it and then thereafter spread subsequently into a given section. You likely have, let me ask actually, how many zones of inhibition do you guys have?
4.
4? How many zones of inhibition do you guys have? Okay. So does everyone have 4 or 3? 4? And 3 for E. coli? Oh no, I'm saying for just a particular, okay, so you have 3 and 1, right? Pretty much 3 for E. coli and 1 for Staphylococcus epidermidis. No?
4 for
both, okay. Okay, so if even we'll say at max 4, right, to make it easier. Reason I say this is so that way you can control and take this into account. That one of those sections will just be your positive control out of the 5, and the rest of the 4, if you have 4, will essentially be used for the subculturing of your bacterium inside the zones of inhibition. Does that make sense? Exercise 2 of last week, essentially. Yeah. If there are questions, let me know. I can walk by accordingly. But I'll say this one last time. Take it from the inside your zones of inhibition, and then subculture them into your new plates to derive new colonies accordingly, or a pure culture. Redarking incubation, yes. For which step? Should exercise 2? I'll answer. So let me answer 2 upcoming questions. To clarify, it's okay to actually take out colonies outside of your zone of inhibition, because technically, based on what we see, if you have resistant colonies, they should be present in your section. Now, regardless of where they're located in that zone or outside the zone of inhibition. I say that because technically speaking, the resistance of those colonies should depict specific characteristics within a finite range. So just bear that in mind. For exercise 2, for the 7 plates, we have lights that is present there. And essentially, the conditions for the plates that you're using that require light incubation will be hitting on the lights here. So don't worry for that time. I would just say, as you see here, there is a group that could actually go forward with exercise 1 for their plates. So if there's a group that's waiting, they can bring in their plates right now.
all groups done exercise 1 except for group 6? Which group are we in? Fourth question. Who is waiting to do exercise 1? Are there any groups that need to do exercise 1? If there are no groups that need to do, if groups are completing exercise 1 and are moving to exercise 2, this is what you need to do for at least light incubation. For the cases where it's dark incubation after adding in UV, if it's based on your specific plate, up to not, do time. For the dark incubation, wrap your entire plate in aluminum foil. You can do it with the dark side, the more tainted side inwards, and then you essentially wrap it up. So that way it mimics dark incubation, and then they'll be accordingly placed, subsequently used thereafter post incubation. The lights will be turned on. Don't worry too much about them for now, but I just want to make sure that. Okay, let me answer other questions in this time. Exercise. So remember, the light incubation is going to be done here. Label as well as the aluminum foil when you wrap it, so that way we know which practical and which group you're part of. Oh, someone's Bunsen burner is too high on. Who else is? Okay, just make sure that Bunsen burners are not too high on.
Yes.
Yeah, please. Does anyone need extra plate materials? Anyone need any extra material? No?
Sorry, before I want to make sure you fix this. What's it? It doesn't need you. You just leave it. Technically, we're gonna put it down there where the light is on.
Okay, fine.
You can after you've done essentially the smearing. Perfect. You don't have to wait for all of them. You can put them now. Just make sure that they're labeled, like you know which condition they are from the exercise.
So this is timing between 6, 5, so 6.
That's okay. Yeah.
Typically, like there's a time.
You can go. Like, don't have to take one. Which means essentially you can leave one without a smear.
Wait, should we do this just because we're ready to do this?
If you have the time,
Okay.
they are down there. Other groups may have them as well. If they are done, I would ask them essentially if they could give it time.
Yeah. Wait.
Can I just walk you guys through? So for those of you that are wondering which for each plate, the different conditions, let me just say them out loud to make it easier. So remember, exercise 2, you have 7 plates. Make sure that you label them accordingly. If you don't have them written down in your manual or in your notebook, I'm going to make mention of them right now. Condition 1 is 30 seconds with UV, with your lid off, meaning just like what we did for exercise 1. We're going to incubate on light and dark afterwards, but what I just want to highlight is the one that's light is the one on the left. The one that's going to be with dark incubation is going to be with aluminum wrapped up around the plate itself. So just bear that in mind. You could do all 3 plates to come, which is 2, 4, and 7 in dark, but essentially I'll go through each of them together. Your first plate is also going to include a mask, and all plates from 1 to 5 will include a mask. So bear that in mind. The star-shaped mask suffices. Now, bear in mind that the difference between condition or plate 1 and 2 is that it has light versus dark incubation. So plates 1 and 2 will be 30 seconds at UV or on UV. Plates 3 and 4 for 3 minutes. And essentially all of those 4 plates will have the lid off, and they'll alternate from light, dark incubations. So conditions, hopefully everyone's paying attention. Plates 1 and 3, they're going to be with light incubation. Plates 2 and 4, dark incubation. Apart from that, the pairs of 1 and 2 will have 30 seconds at UV, 3 and 4, 3 minutes at UV. Plate 5 will be 3 minutes at UV, but unlike the first 4 plates, the lid will be left on. And that'll serve as an additional constraint to see if the effects turn out well. And you'll use it at light incubation with the mask included on UV. The last 2 plates, plates 6 and 7, will not use any UV and will just differ with light versus dark incubation. Once again, light is with the light, the natural or ambient light there. And then dark incubation overnight will just be with aluminum foil wrapped around your plates. So remember, plates 2, 4, and 7, dark incubation. Once again, hopefully everyone's paying attention. If you're stuck, I want to just say it once because it may make it too repetitive. Please bear in mind that you're doing inoculations 3 times with the sterile swab. You just mix once from your agar slant, but make sure that it's viscous, and importantly, that you're doing aseptic technique. Plates 1 and 2, 30 seconds UV. Plates 3 and 4, 3 minutes UV. All 4 plates without a lid, and they alternate from light, and dark, with a mask included. Difference for plate 5, pay attention folks, 3 minutes with UV with the lid left on. Very important. Light incubation included with a mask, and then the last 2 plates without UV. Only difference of plate 6 is with light incubation, and plate 7 with dark incubation, i.e. with aluminum foil. Any questions for the setup of exercise 2? No? I saw some questions or some people asking questions. Any questions once again? Maybe that answered it for many of you. Maybe can I ask for, oh yes, question. Okay, so I saw some escape mask down there. Did anyone not even start getting one? I see them right here. So you guys can make these look like here. So leave that on the sheet. So that way, well, that's here. The 4 will be on this side. Remember that you sign on the start of the PV. Yeah. So let me just say this one more time. Don't the condition, if you're doing section, hearing, take it from inside. Responsive control, take it from outside any section. Should work. But you don't have to take off actually yes. That should run. Any other questions? Yes, it's same. Yes, so you can do it with the lid faced up. That's okay. But the reason as to maintain it for the other ones, now think about it logically. If you were to actually leave it up open, right, you're actually leading to greater risk of cross-contamination with the airborne environment. So technically, for the other cases, plates 1 to 4. Generally speaking, you would not want it to be left open, the lid off, and then left essentially with air exposure, airborne, for bacterium to creep in. Rather, you would actually have the lid inverted or the agar on top. And generally speaking, yes, the UV, you may wonder, okay, like, would it penetrate through? But technically speaking, you did this for exercise 1. And technically, if you think about it, if you left the lid open, you're running greater risk for a bacterium to go through, especially those that are UV resistant. So generally speaking, and if I'm not mistaken, that's how it's been done in the past as well. Yeah, if there is, let me see. I don't think there's any other conditions. Yes.
Would we intubate them upward like last time? Or we invert them this time because we don't have the distance?
Inverting is okay. No problem.
Introduction.
Perfect. Oh, that's these are extra
This is for right now.
results.
Yeah.
That's great.
Thank
No problem. Yes. They'll be left there for the day. No problem. Just to make sure that they're all labeled. Yep. Folks, please make sure for the light assay, like for incubation, bear in mind that you label all plates individually and as your group in the practical. Only after. 1 minute.
Where would that be?
Auto.
Oh, okay.
That one there, though I think autoplug it. Your question.
So you mentioned that if it has like these little lines, that it shows resistance. What does that look like? Like, is it like?
I do it like that. Yes, I think it would be like
Okay. And that means that the bacteria has resisted.
Essentially, the one that I'm there. The thing about these ones is that it's kind of like washing with hot water. Essentially, that means
There was a question earlier that for the extension predict what you will see if the antibiotic is bactericidal or bactericidal. So the ones with smooth edges will be static or sidal. But it was
I actually sent it out.
it's the last thing. That was fun part.
If it was that one or the one before, I said that there is essentially two, in upcoming weeks we'll be having two sets of presentations.
Oh, okay. My bad. But and basically, my friend was in a meeting lab. He basically sent me the question that his team. Okay, I'm not sure if it's his team, but basically he was
Sorry,
talking about the type of antibiotics.
Yeah. So
And like
I don't think we're gonna be doing that for today.
Okay.
But that being said, you are allowed to. It's like, so for context, I send my messages personally to our practical, but some TAs would link to the entire
Yeah.
group. So, and that's okay because it gives you also other interpretations of what needs to be done today. Yeah.
I don't know if that's the same as what we're doing on our presentation. Like, it's strictly just result what you got. Unless going to
I will ask different questions, but generally speaking, I would say go with your results. I'll actually talk about them in about 14 minutes, if that's okay. Yeah. I want to answer a question regarding this, but what is your positive control actually any bacteria?
Pick the one that I don't.
Yep. You can.
And then I do that. What is you said that you can pick inside?
So, technically, okay. It does. Now it just is important to know where you're going with this. Because okay, which one do you want to be from So this is actually a good example. You see very smooth edge, right? And so essentially colonies that would exist here, if they do exist, would essentially be resistant to the antibiotic. And
So they're inside. Right. Okay, that makes sense.
so what you can do, let me just say, because I have like I think about the changes in the long run.
Yeah.
Like as you will probably see next week what the result will look like. So that's why I know it shows certain. Let me just make sure I don't make a mistake. Yeah, go with the ones on the side, and that way you will get. Hopefully, if you see any colonies, they likely are the resistant ones to strep them out.
So here, so in the circle one, are we seeing a bacteria side?
No, I'm going to talk about that.
Okay, because I was just reading. Are static, but
It
then
depends.
there's one that not. So that one, the one with the
Special ones.
that one is both for the five. Is that true? Okay, that makes sense. Okay, thank you.
No problem. Let me answer a common question for some of you. If you're ready, for the dark, actually, outside is really. In the past, so the laptop would actually put them inside. I'm just trying to remember. Inside is okay. As long as I just want to make sure I don't make a mistake because I don't want you guys to have aluminum inside if it's not needed. Let me just make a So, folks, I just want to clarify something. You may be wondering what's the difference between bactericidal and bacteriostatic antibiotics. Please pay attention, folks, because it's very important indeed. If you recall, have a look at the plates that I drew here earlier. Folks, are we paying attention? Group 3, are we paying attention? Yes? And I'm not saying it to the entire group, but for those that are not paying attention across our groups, just bear in mind the following. You will see, based on your Muller-Hilton agar plates from last week, specific patterns or shapes of the zones of inhibition. Some are very smooth, and some are staggered or daggered, essentially, looking like spinal arrangements like the ones here. And essentially, the ones that are smooth depict bactericidal antibiotics, or particularly showcasing a bactericidal effect of that antibiotic for a particular bacterium. So, for example, let's say, this is streptomycin. Streptomycin is here for Staphylococcus epidermidis. Then that means that it has a bactericidal effect, as it shows smooth edges. Whereas, let's say streptocycline, for example, in Staphylococcus epidermidis, shows daggered edges within the zone of inhibition, looking like a ripple around the zone of inhibition. Then that indicates a bacteriostatic effect of tetracycline on Staphylococcus epidermidis. Does that answer questions regarding the difference? Yeah? Because this is something we want to bear in mind for next week when we discuss these details further. Yeah. Yes. That's, so think of it like in terms of yeah, I would say go essentially with what I said: tidal, smooth, static, staggered. Let me answer your question in just a moment. Don't worry, they're saying it for the group. Fraser, how's everything? Everything's the other thing is though, it's just like actually what's your
I just want to know, you know the jaggedness? Did you see any jaggedness on your episode? On the the disc?
I found some of it, but I didn't
Is that like without your you seeing? You know what that is? What that means?
Can you
Basically?
repeat the
You know what that means if it's seeing the jaggedness? Is it,
Yeah, it's pretty much the
it's tidal.
I
I mean, they have to do day 2 anyways.
yes. I would have actually assumed it's static.
Is that why? Because it's
No.
not. I'm trying to see.
I mean, we could also
Well, they're gonna do day 2 anyways, but it's
Yeah.
know. You can't tell until you do day 2, really, to tell
as much, but technically the zone of inhibition should give it indication.
Yeah.
Like and
That's
no,
what I'm
yeah. Actually, have you been telling them to measure it? Yeah.
Just give it a read.
Yeah. Because it's a good point.
No, that's fine.
Yeah.
Before they do that.
Other observations. Yeah.
Okay, thanks.
No problem. No problem. No, thanks a lot. Let me just clarify this one part for them. For you guys, sorry. At room temperature. So don't worry. You can place it. I would recommend down there.
Down there.
Yes.
Okay, thank you.
No problem.
I just wanted to ask.
Is this smooth and is this jagged or is this smooth?
I think let me see. Yeah, actually, I can't remember. We should be the same one.
Yes.
Well,
This is-
okay, we gotta go on.
These two go together.
This is the,
So this one.
yeah.
the jagged,
Yeah, that's fine.
so jagged. Well, we like, we swapped it, so it like messed up the yet. That's fine.
You can also record or observe essentially the range of that zone of inhibition. Some are larger, some are small, some are intermediate in size. You can essentially account for those as well.
Yes, you can swap like where the disc was.
So just around, right?
Okay.
Yeah.
Yeah.
It's actually just touch it, okay. But because like this is, for example, quite large, I would recommend going around the edges.
But not on.
If you had a choice between touching the disc or the colonies themselves, go with the
Obviously,
disc.
yeah. Okay.
That's what I'm. No.
Yeah.
Everything else okay?
These are the ones that get involved.
Put it in there. Oh, okay. Put it in there and they will recording it. Are both of them okay?
Yeah.
These are all equal. Yes. And I would say put one on board and one. Everything okay, folks? Are we done exercises 1 and 2? How long until we'd be done? Yes, you can tape on top of the aluminum foil for determining your practicals group or group number. Just to bear in mind, I want to make sure, is every group done exercise 2, yes or no? Group 5? Is group 5 done exercise 2? Done exercise 2? No? How long do you guys think you need?
Yeah.
You did 30? 10 minutes? What? 5 minutes? Are you guys almost done? 1 and 2 as well? Yeah? Perfect. Okay. That's great. That's great. You can actually. No, for the dark one, they should probably be around. The 4 plates for exercise 2 and like patient together. Please come back to our region because you don't want to find it difficult to read afterwards. Especially if the lab has to hold them all together. For that, make sure that you find out together. Regarding how many groups are moving into last week's subculturing of the antimicrobial susceptibility test, please? You're done? Perfect. Okay.
Are you guys also recording your observations? For your postdoc? Great. Yes. It's gonna be in the incubator in the top left side. Does not have to be placed inside the incubator. Yeah. Great question. Dude. By the way, I can hear you guys when you say questions and like jokes with each other, so don't worry, like, but I have to divert my attention. So yeah. Okay. So folks, how are we doing so far? Um, are done, the MH agar plates from last week. Folks, when your groups are done, the MH agar plates from last week for subculturing, place them inverted back into the incubator on the top left side. Just bear that in mind. Once again, all plates from exercise 1 to 2 to last week are all labeled with the practical, your bench, the exercise number, and the different conditions, as well as the date for the exercise. Yes.
Functions.
What's your question? No, you have a presentation. Talk about your germicides, microplate reading results. So, yes, and so you guys might as well prepare. If you have your phones, you can look on Quarkus, see the results, and be able to talk about it and answer questions when I ask you. Just have a look at time point. Compare at 0 and 120 minutes. Yes. This is clean. Yeah. Jagger. Yes.
This is bacteriostatics. This is bactericide. So A will be decided, T will be decided. This is cluster. This is.
So this, this may be. Yeah, it could be static. It's hard to tell. When your plates are hard to tell for specific results for the discs, I would say based on what you see for subculturing, you'll be able to determine if it was indeed sidal or static, essentially.
Right now we can't tell what it is.
You can to a degree. It's hard to tell actually, but because it's quite faint. This one it will be hard to tell, but I know this one's staggered.
This one. Right.
Yeah.
And it's the same for this one as well. This is being staggered. SK was staggered. P had nothing for both of them. T was very little, and it's come smooth, and C was smooth. It's the same for everything.
This is smooth, but this one is static.
P means that means P is static. That's what I say.
Yes. Well, let's have a look and compare. They may differ. Oh, you know, it may be also hard to tell from the figure itself. It could be because as you took it out, that maybe some of the colonies themselves are out accordingly. So it's very hard. We'll likely see, compare, and we'll hopefully compare the other results as well. But I think this actually moves down.
Yeah.
Yeah.
Okay, sounds good.
Does that answer your questions?
Yes, and now we can throw them. We don't need these ones because we replace it and we are keeping it.
You can, if you recorded them, you can disregard them. Like you can discard them. No problem.
Okay. So I answered the rest of the reflective practice questions, but the only one that I didn't understand was the question. I don't know if you heard what it says. Is it likely that it's sin resistance can be transferred from J to 69?
So essentially, so the colonies, so for conjugation, I would say have a look back to the exercise. Which one has the conjugated plasmid? Okay, actually have a look as to which one, like where is the resistance gene located? And that will help. Potentially that. Folks, I'm gonna just try to get your results uploaded onto the screen to start getting ready for your presentations. So I'm gonna give you guys about 6-ish minutes to discuss as a group, to discuss your results, talk briefly about your intro. Each person should be speaking. And remember, it's timed for 6 minutes. If you could be done in 5 minutes, that'd be amazing. But essentially, talk about your intro. So, okay, if you guys are wondering, let's just step back for a moment. Hopefully, many of you are done all exercises for today. If you're not done, just pay attention with what I'm saying in the meantime. Your presentation is going to be focusing on the germicide microplate reading results. So bear that in mind. From what we did last week, I posted them, uploaded them on Quirtus. You should be able to see them. And your goal is to, one, introduce to me what the project or objective of the germicide experiment was. That's your intro. In about half a minute. Actually, I'll just go through the details now. Your results includes what your conditions were from your group, and I want you to tell me what your trends were from 0 to 120 minutes, i.e. from 0 hours, or like it shows on Quirkus 14 minutes, all the way to time 9, your 9th time point, which is 2 hours and 14 minutes. So essentially, I want you to give me, describe to me what your results appear to be, and then thereafter be able to share with us what you conclude from the effects of your antiseptic or chemical germicide on your bacteria, be it Staphylococcus or E. coli. Then you could briefly talk about, for the last portion, any limitations or extensions that you could provide. And so within that framework, try to be done your intro in a minute, your results in 2 to 3 minutes, and the remainder about 2 minutes or less for your discussion. Any questions, folks? The goal is that you want, and it's okay, you could use your notes, you could use your manual, your notebook, but just try to be engaging and be able to describe what you were able to observe and introduce the concepts or conclude accordingly as members of your group. I do want to also say, actually, yes, I'll answer questions now. What's your question? Yes. No. Yes. Yes. Give me a moment. I'm gonna have to upload this in a moment. Yes. Um, yes. You guys are done? The trans. None of the antibiotics. Let me clarify. Okay, so I'm gonna just share with everyone. To clarify, folks, you can talk about your antibiotic results if you want as well. Antiseptic is what I want you guys to focus on for now. But hmm, um, I'll ask questions when needed for the other portions. Pretty much what Dr. Brunt wants me to have you guys talk about is your results from last week up to the results you gained from this week for antibiotic susceptibility test. So just, um, okay, this is what you guys can do to make it easier. Introduce to me in 1 minute both exercises, the antibiotic susceptibility test and your antiseptic germicide essentially experiment. Then talk about your results, 1 minute for each exercise, very briefly, and then the conclusions from each result, each experiment, followed by limitations or extensions that you could provide. And that should suffice. Do it for both experiments, and that should be okay. By the way, anyone's Bunsen burner still on? I smell something on. Yes, yeah. Okay. We'll start in about 3 minutes. 2 to 3. About 3 minutes. So look at your column. Everyone's Bunsen burner still on. Is everyone's place in the incubator or down there with the laboratory stations? Yes or no? Does anyone need to use the Bunsen burner? Are all doors closed for your exit? And then talk about it. Okay. May Allah subhanahu wa ta'ala accept all your efforts in this session and the next. Subhanallah al-Azim. ورب العرش الكريم لا إله إلا أنت سبحانك إني كنت من الظالمين. I'm not sure how far the subhanallah. Inshallah ta'ala, I just wanted to highlight Bismillah ar-Rahman ar-Rahim. أعوذ بالله من الشيطان الرجيم رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. Briefly, students went over the antimicrobial susceptibility test. They did presentations, they spoke about their results and connected it back to the germicide results that they did for their specific group. Thereafter, and before this, they looked into the UV. They did tests, and I talked briefly about the generation time assignment to remind them as to what their next steps need to be pre-submission or post-lab. And so I really hope students were able to actually connect the dots between different unique exercises and approaches, as you can see, like conjugation and transformation, quite readable to many upcoming or previous exercises as well. And even when we talk about processes like using germicides and antibiotics and the biochemical, connections are things that really connects us, uniquely. And subhanAllah, just imagine how, like, for example, if we look at conjugation and put two different organisms together in the incubation, like E. coli and Staphylococcus epidermidis, then we, test the same results we did for the microplate reader for a germicide incubation, then maybe we come to appreciate results accordingly. And inshallah ta'ala appreciate them thereafter. Barakallahu feekum. Subhanakallahumma wa bihamdika nashhadu an la ilaha illa anta nastaghfiruka wa natubu ilayk. Assalamu alaikum wa rahmatullahi wa barakatuh.